My name is Enrico Novelli, and I'm a hematologist, so I'm an expert in blood diseases. And my main area of focus is sickle cell disease. So I treat patients with sickle cell disease, and I do research in sickle cell disease.
Okay, and how did you end up pursuing hematology of all things?
It's a fascinating field where for a long time we've known that progress in the understanding of the human genome and human genes, as well as transcriptional regulators, has really bore fruit in terms of development of new treatment. So it's an exciting field where there's a tremendous progress. Specifically, sickle cell disease is a wonderful disease to study from a scientific standpoint. And in fact, we owe to sickle cell disease a lot of progress in science over the course of a century since the disease was discovered. Perhaps more importantly, it's been an orphan disease for a long time, where we haven't had enough funding for the disease. The disease carries a tremendous burden in terms of impairment of quality of life, and a lot of people suffer without having access to medicines or not having enough medicine. So it's an important area to work in, and where anybody can make a difference by studying the disease and applying themselves.
And if I understand right, you're not just a researcher, you're also a clinician. So you see patients on a regular basis?
Yes, I do see adult patients with sickle cell disease. I'm an adult hematologist in the sense that I completed internal medicine training and then fellowship in hematology oncology. And on the other hand, there are pediatric experts who see the children. And there aren't many of us around in the United States, unfortunately.
Wow. So what's the benefit of being both a researcher and a clinician when you're trying to make an impact on a disease?
Well, it's always important when you study a disease from a scientific standpoint to remain rooted in the clinical aspects of the disease. In other words, what does actually affect the patient? What does the patient care about, right? Because you may study certain things that may be fascinating but have no bearing on the life of the patient. And one thing we notice is that we now have more and more people getting old with sickle cell disease, right? It used to be a disease that used to be fatal in childhood. And nowadays, in developed, in high resource settings, the majority of people survive to adulthood. But with that comes a burden of complications, right? The longer you live, the longer you are exposed to this terrible disease that affects your blood vessels. And the damage to the blood vessels results in a damage to the organs. And among the organs, we see that the brain is very affected in the disease. And so what we find is that people who age with sickle cell disease tend to develop a type of cognitive impairment. In other words, an impairment in memory, an impairment in executive function, so the ability to perform complex tasks is impaired, the ability to recall information. And this is very detrimental to the patients because they have a hard time remembering the medications, remembering the appointments, and following through with instructions given by their doctors and healthcare providers. And so it is a great burden of disease. And so that's why this has been a major area of focus for me and my team.
Wow. So let's talk about the research. I think you've mentioned that cognitive impairments can occur in early stages of the disease as well. In some of the Alzheimer's work that Sickle Catchers has supported, we understand that some cognitive symptoms may be more reversible in earlier stages of the disease. Do you think that's also true in this case? Do you think if you see that there's physiologic evidence of potential cognitive impairment that might not be coming out yet, people aren't showing any problems on tests or anything, that potentially that's reversible if you figure it out?
Well, in fact, thank you for bringing up an important point, that sickle cell disease is a disease where there is accelerated aging, accelerated deterioration of organ function. And so we see people with sickle cell disease in their 50s who have organ function that you would expect to see in an elderly person. And the brain does not escape this paradigm. And so we do see accelerated brain aging in sickle cell disease. And we do believe that, like many other aspects of the disease, this is preventable, or at least it could be slowed. Right now, we're in a situation where we don't have any biomarker, in other words, any laboratory investigation or any imaging study that can tell us this person is actually somebody who in a few years will develop a cognitive impairment, and you have to intervene to prevent that from happening. Unfortunately, we don't have that. We just have a cognitive test that at some point we find to be abnormal, and we don't know how that patient got there, right? Was this something brewing for many years, or was there an insult, in other words, an injury that caused this? So we do think, though, based on what happens for instance, in the kidney, we do think that the deterioration is progressive as the patient's age. And so we hope that we can find signs of this deterioration early on, so before the deterioration becomes clinically apparent, before the patient actually tells you, I can't remember things. That's too late, right? I mean, I'm not saying that we could treat that person and make them better. That's possible too, but we always want to prevent a damage from happening if we can. That's what leads to this research that we've been doing in partnership with our colleagues in neuroradiology, neuropsychiatry, the School of Public Health, and our colleagues have expertise in other diseases like Alzheimer's. So they've helped us adapting their techniques and their studies to our younger population.
So you're collecting, you have this fancy equipment. It's like an MRI on steroids, right?
That's right.
It's the 7 Tesla, so that means you get very high resolution images, and that allows you to see something that previously couldn't be seen. I wonder if you could talk about that and what that has to do with what you're trying to figure out.
Yeah, so I'm not a physicist or a neurophysicist, so I can give a simple explanation, but as MRI technology has progressed since the 1980s, since the technology was invented, the resolution of the images has improved, and that is dependent, among other things, on the strength of the magnet field that the MRI equipment uses. And the unit of measure of the magnet field is Tesla, and so in clinical use, we use 1.5 or 3 Tesla machines, but for this research, we've used that 7 Tesla machine. And what that means is that we now have much better resolution, and we can image small blood vessels and small structures of the brain better than we've ever been able to do before.
And having this very high resolution imagery of the small vessels, what does this have to do with sickle cell disease and cognitive impairments?
Yeah, so sickle cell disease is a disease of the vasculature, of the blood vessels. So the damage starts in the red blood cell, and the red blood cell travels through blood vessels. The damage to the red blood cell is the spark of a fire that ignites the vasculature, ignites the blood vessels, causes a inflammation like you would see in somebody with an infection, but there's necessarily not an infection. It's what we call a sterile inflammation. And inflammation causes the expression, the production of a lot of inflammatory molecules that cause a damage to the blood vessels, in very simplistic words. So there's ample evidence that the blood vessels are at the core of the disease, of the pathophysiology of the disease. In other words, it's what causes the organ damage. Right? There's a damage to the blood vessels, there's not enough oxygen delivered through the blood vessels, and there's a damage to the organs. And so we believe that cognitive impairment stems from a damage to the blood vessels. Now, we know that children with sickle cell disease have a damage to the large blood vessels, and that will cause a stroke. But we also know that people without damage to large blood vessels that cause a stroke also have cognitive impairment. And so now we're focusing on the small blood vessels, and we do it also because we can, thanks to these new techniques, MRI techniques.
So I just want to make sure I understand this, because I think I just learned something new about sickle cell disease that I didn't know before. Correct me if I'm wrong, but I think what you're saying is that this hallmark characteristic of sickle cell disease, which is the shape of the red blood cell that looks kind of like a crescent moon, when the red blood cells of that shape travel through the vessels, that somehow that causes inflammation of the vessel, that is the shape of them that's physically somehow creating inflammation?
So you could argue that the conformational change of the structure of the red blood cell from a donut without a hole, which is the normal shape, into a crescent, which is a sickle cell, is the fundamental lesion of sickle cell disease. And that's for 2 main reasons. One, because once the red blood cells assumes that form, it becomes more prone to destroying, to breaking up in the vasculature, and that's what we call hemolysis. And when the red blood cell, so the red blood cell is a bag that contains a lot of important stuff and contains a lot of proteins that could be damaging if outside of the bag. And so when the red blood cell breaks up these molecules of it that we call damage-associated proteins. They are now free-flowing in the circulation and cause inflammation. That's one issue. The second issue is then when the membrane of the red blood cell becomes deformed, it expresses on the surface molecules that make it sticky to the. So it's, in other words, the red blood cell becomes adhesive. It sticks to the blood vessel, and when that happens, of course, then it tends to recruit all the cells of the blood into the blood vessel, and so you have areas where there is a log jam of red blood cells. So imagine if a river where there are a lot of logs, right, that free flow into the river, but there are areas where the banks of the river are damaged and the logs get stuck. So that's one way to look at it. Or perhaps you have logs that tend to stick a little more to the blood vessel.
It's quite interesting because, I mean, as you know, in stall catchers, we're looking at blockages at the capillary level that are ephemeral. They last, 10 or 15 seconds. And it sounds like you're describing a kind of a stalling effect that can occur on a larger scale with an aggregation of these things getting caught up because of damaged blood vessels and the shapes and blood cells opening up and releasing things that cause inflammation. And it seems like nowadays there's a lot of interest in inflammation in general, that it seems to be implicated in so many diseases, and particularly in dementia.
That's right.
It's like you can't talk about dementia without talking about inflammation.
And that's one of the reasons why we borrow the techniques from the much more established field of dementia and in general cognitive impairment in the general population to sickle cell disease, because there is there is a shared component of neuroinflammation that we think is critical to understanding this disease.
And you made a really interesting discovery. It sounds like you have a lot of the 7 Tesla data, and you looked at part of it, and already from just that small portion of the data, there's some interesting finding. Could you describe what that is and why it's important? Yeah,
we found that in certain specific areas of the brain where we know there is much more susceptibility to damage, hypoxic damage from lack of oxygen or other types of damage. We zeroed in on those areas, and we found that blood vessels that normally in a person without sickle cell disease are easy to see and recognize by 7 Tesla MRI, with the MRI we've been using, are much harder to see in patients with sickle cell disease. And what we see is that these blood vessels that normally would have a nice elongated shape are now fragmented, they're broken up, they're more tortuous, and we think that that's evidence of a damage that is occurring in that area to the blood vessels. We found that having damaged blood vessels was associated with having a damaged cognitive function. So we think we have identified a new biomarker, a new marker, essentially, a neuroradiological marker of cognitive dysfunction. And like it happens for all other organs and diseases, the damage you see by histology or by imaging often precedes the clinical phenotype. In other words, you have damage to an organ or a blood vessels before you actually have high blood pressure or stroke or heart attack, right? So, and we have a lot of ways to detect if somebody has a damage in a coronary artery that we call stroke, right? You do a cardiac catheterization, that's a very standard procedure, and that patient is going to have a heart attack, so we intervene. We don't have right now a test that will tell us that somebody will have cognitive impairment. We think that if we look at these blood vessels, we could have that test. In other words, we will be able to tell that person, look, your blood vessels are not looking healthy. We're going to do this, for instance, more aggressive transfusion, or we're going to use a new drug that can work for the brain. We're going to do exercises to maintain your cognitive function and perhaps recruit other areas of the brain that could help overcome those deficits in those specific areas.
So it sounds like you don't necessarily have this biomarker. Is that right?
Correct.
So some do and some don't.
That's right.
If this turns out to truly be a biomarker that's kind of like a telltale of trouble yet to come for cognitive impairment, if I were a sickle cell patient, I could come to you, you would run a scan, you would see possibly that there are these biomarkers, there's something going on with the blood vessels in my brain, and you could say, you don't seem to have any cognitive impairments yet, but this tells us that you might be more likely have
That's right.
these. And because we know this, we can actually potentially do something now to prevent or delay the onset of those symptoms.
That's correct. That's exactly what we would like to be able to do.
Yeah. It's really exciting, and it's a great explanation that you gave. I appreciate it. So one last question. I know you're short on time. What does this have to do with Alzheimer's disease? Is there anything we can learn from this that might also help with understanding, preventing, or treating Alzheimer's disease?
Well, I think that every time you identify a new biomarker, you not only have a tool to do early diagnosis or for precision medicine approaches where you can tailor a treatment to a patient with a specific biomarker, but you also shed light into the pathology of the disease. And again, there is a shared common pathogenesis in a lot of diseases, not just Alzheimer's, for instance, multiple sclerosis, where there's a neuroinflammatory base. We hope that we will then mechanistically be able to understand more about how these diseases lead to cognitive. And absolutely, I think that studying rare diseases has been frequently yielding benefits for even health controls or people with other degenerative or neuroinflammatory diseases.
So I said that was my last question. I lied. I actually have one more, and I promise this will be the last one. So we're here at the University of Pittsburgh with you in a kickoff meeting for the Sickle Catchers project, and I wonder if you could speak to why we need to have a project like Sickle Catchers. How is it that Sickle Catchers could potentially help you solve this specific research problem that you're tackling?
Well, I think for a number of reasons. One would be, I think it's the computing power is something that is always problematic, right? These are very tedious analysis to do on MRI scans, and they require many hours of work and computers, and so they're very complex to do and very time consuming. And so having the computing power and the human resources that cell catchers or sickle catchers has is a great advantage. I also think that once you recruit a community into a project like this, you create momentum in the community to raise awareness and interest for this disease that definitely deserves to be more front and center in the public discourse in the United States. So I think that there's an added benefit in that sense as well.